Welcome everyone to this month's ReproNim webinar. We are super excited to have Keith Bush with us to share his wisdom and experience in implementing best practices for tools for enhancing rigor and reproducibility in neuroimaging. He's an associate professor at the Brain Imaging Research Center in the Department of Psychiatry at the University of Arkansas for Medical Sciences. And without further ado, I will give the floor to Keith.
Okay, thank you for having me. This is a real pleasure to do this. And I guess I'll just jump right into it. So I guess the big caveat today I want to talk about is there's actually a manuscript that's the premise of this talk. So our group actually published a perspectives article, which is like an op-ed piece for Frontiers, kind of documenting this transition process that we made. And just to start off, since unfortunately the perspectives article format in Frontiers doesn't actually have an acknowledgement section because of the format of the manuscript, the paper that this talk is based on, and this talk is really the result of encouragement we got from Dr. Marianne Martone at UCSD. I had actually reached out to her while we were doing the transition to ask her some questions about data dictionary design. And when she heard the project that we were undertaking, she actually encouraged us to write it up as a case study. She thought it would be useful to the community to see how a relatively small lab thought about the process and kind of how we implemented the transition. So all of this really emerged out of her encouragement. We really appreciate her leading us down this path. And so with that, I want to talk about the goal of this talk. So I want to talk to you. So the paper's available. You can see really the nuts and bolts and the resources that are that we put together to kind of show people how we did the transition and what we thought about the cost benefits of the transition. But what I want to talk about here, since that paper's available to read, is some of the stuff that we didn't put in to that manuscript, kind of our thought process about what does it take to get a lab to try to do this? Like what was our tipping point? And then what were our concerns? How did we find information? Because we really went from zero when we started this process. We didn't know anything about open science other than we'd heard the word and we knew it was good, but we weren't doing it. So part of this talk, or at least a big chunk of this talk, is going to be kind of accentuating the paper. I will give some of the backbone of the paper just to show some of the details of how we did the implementation, but really I want to provide more of the context. So before we do that, let me talk about the scale. So it helps frame how we're thinking about it. So our lab, we're at the University of Arkansas Medical School. And so this left panel here is kind of the main hospital building of UAMS. And we actually have the Psychiatric Research Institute, which is the psychiatry department essentially, is a wing of this main hospital. And the imaging center basically occupies the basement of that wing on the right there. And we're a small lab. So we have one scanner. We just got a brand new Siemens Prisma we're very proud of. And that's basically the picture of the first time the installation crew said it was completely installed and we could look at it. So I took a picture that day. We're very proud of this device. And just to give you some scales of our lab by the numbers, just to say like, how big was this transition in terms of personnel, in terms of resources, in terms of our funding base load? Let me give you an idea. So our personnel, our imaging center is comprised of 6 full-time faculty. Myself, I'm a computer scientist. So I'm the computer scientist. We also have an MRI physicist. We have 1 MD PhD. We have a clinical psychologist who does child psychology and 2 addiction neuroscientists. In addition to the core faculty, we actually have 4 intensive external users from various departments. Like we have an audiologist that's involved. And our core infrastructure beyond PIs is we have 1 program manager. We have 4 full-time research assistants that are professional research assistants, not trainees. And then typically in the lab, we'll have between 1 and 2 trainees, typically postdocs, but right now we actually have a pre-doc in the lab. As I said, we have a Siemens Prisma. We also acquire data on a very extensive psychophys rig that's MRI safe. We have kind of all the standard psychophysiology modalities. And we just acquired a FNIR system this year to help our child psychologist do her research. She wants to actually do some dyadic imaging. And To give you an idea of our compute capabilities, which are very relevant to this discussion. So we have, for the size of our imaging center, quite good compute facilities. We have essentially a processing server with, it's actually 3 servers, but very nice number of cores and RAM, as well as a deep learning server that we purchased recently that was designed essentially to consume the ABCD study, to do deep learning on it. And then also as a side project, to give you an idea, we're actually migrating all our compute facilities to the cloud as these servers age. So one of our long-term projects is to have all of our compute capability and storage capability online. And then to give you an idea of the kinds of funding that we have in our lab. So in the last 5 years, our mix has been, we've had NSF funding, multiple NARSTAD Young Investigator Awards, multiple primarily NIDA R01s. We have a very large NIDA T32 grant that we administer out of our center. We have VA funding. We have a lot of collaborations with the VA. We have a VA hospital adjacent to UAMS that we work with extensively. And then we have a lot of private foundation money, as well as we were just named to be an HBCD study site. So the HBCD is the successor to the ABCD study, starting in even younger children, kind of prenatal and then very early childhood. So that's kind of the scale of the lab that I'm going to be discussing that transitioned throughout this process. So the origin story, why we started this process, it really emerged before this event. We weren't thinking about open and reproducible science at all, other than we'd heard about it, but we weren't really motivated to do it in any way. But really started the process for us was the Eklund et al. Paper that came out in the spring of 2016. When this paper came out, we all read it. We had read it within a couple of weeks of it being published. We're pretty responsible methodologists, so we follow the labs on with the Google alerts that are doing kind of all the cutting edge methodology validation. So we follow these people. So we were aware of this paper early on. We read this paper, we discussed it internally in the lab. We were concerned about it, but it wasn't a concern that raised the level of doing anything. It was kind of one of these, hey, this is serious, let's keep an eye on this, but pretty much business as usual until there's some guidance about what to do, really. And that was how we operated that summer until, if you remember, the takeaway is the gist of this paper, if you're not familiar with it, was based on the way we were doing multiple comparisons corrections for our regions of interest. You could have up to 70% false positives, especially if you're using AFNI, which is our core imaging processing tool that we use in the lab. So there was some issues potentially with AFNI having an internal bug. And so this was alarming, but it unfortunately didn't meet our threshold for really seriously thinking about doing anything. And that really changed in the end of the summer when the New York Times published a mainstream news article in the Sunday New York Times, mind you, discussing this scientific paper in the mainstream. And this coincided with a number of other media outlets making similar kinds of articles, kind of discussing the problems we have in imaging with reproducibility. And the outcome of these mainstream articles was I started receiving emails and phone calls from family members, from friends I went to high school with, asking me if we should trust imaging science. What did I think about it? Is this the end of the field? And I didn't really have good answers other than, well, let's wait and see. This is just one paper. But it started really, that New York Times article really started making me wonder, how do we know that what we're doing is correct? How do we know our science, our methodology, is correct, that we're not just modeling noise, that we're producing real findings? And I started thinking about it a lot. I started reading a little bit about it. And I started discussing my wife. So my wife is a cancer biologist, and it also was starting to appear on her radar. There's issues with cancer biology, problems with reproducibility. There's a big problem with the use of Westerns, not just in terms of maybe like false positive, but also there's some issues with using Westerns fraudulently to justify results that don't have good outcomes. And then there's obviously the famous science paper talking about psychological science. So there's a lot, this issue of concern about reproducibility, rigor is popping up kind of all over science, largely, because, decades of incentivized research that's maybe not focused specifically on getting these things correct. And so I just want to run through, before I talk about the actual nuts and bolts of what we did, this was really the decision timeline. This is like a small lab kind of on our own, coming to the point where we really thought we needed to do something. So we read the Eklund paper in spring of 2016. The New York Times paper was the end of summer of 2016. But what really, we were concerned, but what really jogged us into action, me specifically in action, was we got our first manuscript rejected based on that paper. So one of the peer reviewers was like, you can't publish this because of the Eklund paper. And then just a couple months later, we had two other, they weren't rejections outright, they were major revisions, again, specifically citing methodological concerns raised by the Eklund paper. And the problem was at that time that there wasn't any manuscript out telling us what to do methodologically. So there was alarm, concern, fear that we weren't getting manuscripts published, but we didn't know what to do. And that all was resolved at the end of April in 2017 when Bob Cox's group finally published a paper. They had reproduced the Eklund paper's findings, and they had made suggestions about how to do the correction so that the false positive issue wasn't there. And so that resolved the immediate crisis, and we could have, if we wanted to, kind of gone back to business as usual. But I had had enough of sleepless nights, kind of introspection, thinking about what are we doing? Is this correct? So I decided to do something about it. And what I ended up doing was that the next month I petitioned our center director for funding. So the UAMS is a soft money institution. So pretty much anytime you spend money doing research or anything else, you have to justify it with a budget center, essentially. So I petitioned Clint for some FTE time to really explore open and reproducible science in depth, understand what's the state of the art, what could we do if we wanted to make a move. And he, thank goodness, agreed to this money. And it was directly out of his endowed chair, actually paid for it, to allow me the freedom to read and to come up with a plan. And in July of 2018, I actually released, we did a presentation to the entire imaging center where I kind of summarized the blueprint that we needed to follow moving forward to transition to open and reproducible best practices. And I actually dug up just to show you. So this was the title slide of that presentation. I took a screenshot. So on July 18, 2018, we presented this to the entire imaging center, all the PIs and the external PIs, everybody was in a room, and I talked over the issues, I talked over the tools. And really what I'm going to do for the rest of this talk is kind of lay out kind of the backbone of that blueprint and then other things we learned about executing that blueprint in practice. So what this blueprint, I won't go into the details of it, basically laid out was we need to have laid out the resources that we need in order to do the transition. And it focused on the workflow of the lab. So all of the pieces that go into producing science in our imaging center. And then what I really focused on was conceptualizing how the tools and theories and all the research that's gone into open and reproducible science, by the ReproNim investigators, how those tools could map onto our workflow. And then another big part of the plan was prioritizing the ordering of the mapping process if we were going to execute. And specifically trying to come up with cost benefits, primarily for Clint, our director, to make a judgment on whether or not the money would be well spent. And so he did agree to it. So he actually, again, directly out of his endowed chair, which is, I think, a real testament to his vision, he agreed to fund a team of people to execute this blueprint. And so originally, on the left, that's I don't know if my cursor showing up here. On the left, it's me. So I was basically in charge. I was part of the data team, we called it. Constructing, because I'm the computer scientist of the group, I was basically in charge of building all the software infrastructure to map our data, to use the tools. So some of these are very computationally intensive tools, as well as training 2 of our 4 full-time research coordinators in programming, Python, as well as the data tools that are necessary to do this mapping process. So Laura Spell and Debbie Hodges are 2 of our research coordinators that agreed. They're very interested in learning these techniques, and they actually also got FTE release time to work on this project. Megan Calvert joined our group in 2018, 2020 as a postdoc, and she essentially enrolled in the ABCD ReproNim class. I actually enrolled in the class as well, but I didn't really do it. I watched the lectures, but she actually enrolled in the class, did all the homework, was part of the project week. She was involved. She very much bought into the whole open and reproducible science approach. And so she's really kind of helped me kind of in the later stages refine the architecture and the software that we were building to kind of perform this transition process. So she's been integral, and I think some of you worked with her, so you know her by name. She's really great. So that was the team that we created to do this process. And so let me talk about the workflow, there's nothing really special about our workflow. But this is how we visualized it. So our group is kind of a standard science workflow. We form hypotheses, we construct experiments to test these hypotheses, we collect the data, we analyze the data, and then we report. And it's very standard scientific method. How we thought about open reproducible science, though, is by reading the literature. So I spent a year between 2017 and 2018 reading literature, and kind of conceptually, I was trying to figure out a way to kind of get my head around all the things that are available. And I conceptualized kind of 4 areas, and that's kind of how I talk about them both in this talk, as well as the paper that I cited that we published on this. We think about it as 4 areas, and these 4 areas can kind of map onto a scientific workflow in nice discrete chunks that can be then executed sequentially by a small team. So we think about open and reproducible science as best practices that they're kind of comprised of 4 tool sets. And these tool sets are pre-registration, open science, reproducible science, and fair practice. And again, I'm not claiming that this is how you should think about it at a very high expert level, but this was how a small lab conceptualized it for execution purposes, kind of clustering a lot of papers and tools together under these groups so that we can map. And we really think about the transition process as these 4 tools interacting with our workflow. So the first thing we thought about was how pre-registration primarily interacts with our workflow, and it was largely with experiment design. So when you are thinking about the experiment, you should be thinking about registering the experiment up front. And so we explored tools such as predicted, open science framework, looking at the differences between pre-registration and registered reports. All of that was constructed around the thought that if we're going to execute, we're going to execute at the point of experiment design and make that clear in our policies that PIs needed to implement those practices at experiment design phase. We think about open science as a set of tools that interact with our data analysis and reporting segments of our workflow. So we thought, again, this is how we were thinking about it internally. We thought about open science really as transitioning to open source code for doing our programming. We were actually using MATLAB originally, and not for any good reason, not because we were using powerful proprietary tools like Simulink, just because it was a language, it was a scripting language that was some of the people in the lab had known. And so this kind of was in all of our code stack. And so one of the primary missions was transitioning this proprietary language to an open source language like Python or R. We also thought about open science as doing things like repositing the code, not just for version control reasons, which are good software engineering practices, but just be able to share our code with our reporting when it was done, and sharing it with collaborators and whatnot. So putting the code on GitHub or some other service like GitHub. We also thought about open science as pre-printing all our manuscripts, like before we go to peer review, pre-printing. When we do publish, we select open access journals or journals that have kind of those green, gold open access tiers, as well as, and kind of the final step we think about open science is putting the data, as much data as possible, online. And there are issues with that, with consenting, but we were thinking if we're going to implement open science, it's really about trying to execute on these 5 areas as much as possible, given where we were. We think, and I know this might be the one that's the biggest divergence from this group. So we really limited our thinking about reproducible science to containerized pipelines. So I am aware that, reproducibility is a much bigger issue. There's a lot, all the way up to experiment design when you're thinking about power and whatnot. But for the purposes of this transition, we constrained reproducible science to be, how do we at least once we've migrated our code to open source code, then how do we make sure that when we utilize that code, we're using the same libraries every time? When it executes on different software platforms, it executes the same way. And so we spent some time looking at containerization, what's available in containerization, how containerization works. Again, I have a computer science PhD, but I went to I graduated 2008. Most of the tools that are involved in this were developed after I had graduated. So it was a learning process for me as well. I wasn't using any of these tools. I wasn't familiar with them. So, I had to go back and kind of learn a lot of what had happened in kind of modern software engineering in the last 10, 15 years. So that was a process for me. But that's how we thought about reproducible science. And then the last piece, FAIR. So we think about, FAIR, again, you're all experts at findable, accessible, interoperable, reusable data practices. And these are brilliant, really well specified set of principles. But we focused specifically on the aspect of FAIR that we thought would have the biggest impact and was most relevant to our work, was the use of data dictionaries. Constructing data dictionaries, tying, identifying ontologies that were relevant, identifying elements of those ontologies that map directly onto the data that we were acquiring, finding these numbers that are persistent and publicly available, so people semantically know exactly the data elements that we're collecting, and these data elements can be shared with other groups to do collaborations, can be attached to our data when we reposit it, so that future individuals, largely we were thinking in terms of future graduate students and postdocs that come in our lab that want to work on older data sets, they would exactly understand what those data elements that are in the data that they're using, what they mean, the reason they're in the data set and whatnot. So we realized there was a lot of advantages of using FAIR just for curating our own data so that it's easy for us to understand our own data, both now and in the future. So that was kind of our overall thought process about how we could extract from all of the work that's been done in ReproNim, boiled it down to these very digestible units, map it onto what the work that we were doing in ways that we could really clearly understand how they would map. And then we started thinking about, well, actually, how could we execute? And after thinking about this entire ecosystem for a year, it dawned on me, and I guess this is obvious, but when you don't know anything and you're kind of self-teaching domain, it dawned on me that the real key to unlocking open and reproducible science is the data standard. So everything revolves around standardized data. And so that was when we focused on BIDS. So this is the brain imaging data structure standard. I believe this came out in 2016. I think that was the first paper, if I remember correctly. And so BIDS is kind of a standard way of organizing imaging data, in terms of naming conventions, in terms of directory structures, and it has so many advantages. It was really shocking to me when I thought about how this could unlock all of these other tool sets. So for one, we realized that once we got our data into BIDS standard, we got reproducible science for free through BIDS tools. And again, these are low-hanging fruit container. I'm not talking about containerization of detailed statistical analysis, for individual manuscripts. I'm talking about containerization of the bulk of the processing in the lab, just going from raw to minimally pre-processed data, like trying to lock that down as reproducible as possible. So by mapping to BIDS, you get BIDS tools. By mapping to BIDS, you also get OpenNeuro. So OpenNeuro is essentially this online repository that if you have a BIDS formatted data set that's validated, you can just easily upload that data to OpenNeuro, and you now have public repositing. So those tools just are free. We don't have to do any work as long as you do the work of getting it in BIDS. And then finally, we realized that the JSON sidecar files that attend all of the imaging data in a BIDS map data set, that is essentially the tool that you use to integrate FAIR, right? So that's where you put in your data dictionary. So in the JSON sidecar file for data element, you basically say, hey, this data element's, I forget the exact key, but essentially label it as, this data element is linked to this ontology with this unique identifier. And, here's like the DOI for it. And so you can easily insert the semantic meaning of the data through the JSON sidecar file of the BIDS standard. So once we kind of figured all this out, it was very easy to think about a process moving forward where we could make a pretty effective transition in a time that we can, kind of a time and an effort that we as a small lab could afford with the level of expertise that we had on site. So all of those pieces working together, we thought this was the plan. So this was what we were trying to achieve. And I know there's a lot of other things that you can do with ReproNim, but what we mean by a transition was executing all of these things, getting all of these pieces of the puzzle working as effectively as possible. Okay. And just to give you an idea of what we achieved. So in July of 2018, kind of the list of bad practices on the left. So in our lab, and I'm kind of ashamed to admit this, but this is really the state of our scientific expertise in 2018 was we had never pre-registered a study ever as a group. We used MATLAB, a proprietary language, for all of our software stack. Everything. It was other than kind of individual, processing commands, which we primarily used AFNI. We didn't track any of our software. And again, GitHub did not exist when I was graduate school. So we had SVN, but we didn't really use it because usually you were working on code as an individual, and kind of group collaborations is an industry thing, not an academic thing. So I didn't have any experience using it. I didn't use it. We didn't reposit any data online. That's not exactly true. We had worked with Enigma a little bit, but other than kind of supplying Enigma with a small amount of data, we had not done any kind of curation. We didn't even know what a data dictionary was in 2018. We never pre-rendered a manuscript. We had a couple of open access publications, I think 2+1 publications in the lab, but it wasn't for the purposes of making it open access. It was just like the right manuscript in the right level for that kind of dissemination. And we didn't know what containerization was for imaging at the time. Again, 2018, this is pretty recent history. And we didn't do any kind of automated QC or anything. Our quality control lab was essentially a summer interns we trained, or not summer, interns in general we trained to kind of visually inspect data, looking for artifacts, looking for registration problems and whatnot. So that was the state of the art in 2018. By the end of the process, 4 years later, so we considered that we had completed our transition process March 2022. We not only pre-register every manuscript, not every manuscript, every study that we're working on, we actually have an internal policy where we budget in our grant proposals for a burn-in period for pre-registration. So every aim, let's say in an R01, has to have an appropriate amount of time budgeted before data collection where we actually pre-register, and we put those in our budgets. It's typically 6 months, 2 months per aim to burn in to make sure that we've registered the aims up front. We have completely migrated our software stack to Python. We now curate all data in the lab, either through GitHub or Data Lab, depending on the group that's working. All of the data for which we had consent authorization to do so has been put online on OpenNeuro. We didn't have the correct consent authorization, so we've actually altered our consent process to make sure that the language is in there such that we can release the way that we want to release. We now have a lab-wide data dictionary. And I actually want to take it aside here. Of all of the things that we did in the process, the only thing that we would have not achieved, we would have not achieved data dictionaries if COVID had not happened. And I can talk about that a little bit at the end when I talk about cost benefits, but essentially from our chairs, the data dictionary process was such a huge burden in terms of what it would take to make it possible. The only way we actually could find the resources to do it was when the pandemic hit. Our imaging center, like probably all imaging centers, was shut down. We were only shut down for a few months. We actually got permission to start acquiring again by the fall of 2020. So we were only down like 4 or 5 months. But during those 4 or 5 months, we had 4 professional research coordinators that were essentially full-time employees getting paid that had absolutely nothing to do. And so we used those 4 or 5 months to train them on data dictionaries, train them on ontologies, and then we actually set them off in building an ontology for each individual study that was going on in the center. And then once they had actually build a data dictionary using ontologies, kind of formal processes for each individual study. And then we had a merge process where all the coordinators came back and we kind of adjudicated differences and semantic meanings for different data elements, because most of our studies have shared elements in terms of the assessment instruments and whatnot. So we actually kind of adjudicated what is the right semantic meaning, and we came up with a standardized data dictionary that covers all the studies going on in our lab, that we now share across all the PIs. So like everybody has access to this data dictionary. And so when we're assigning elements, we know exactly what numbers we're going to assign to data elements. And we have a process in place. If someone brings on a new instrument that isn't part of our initial, part of our kind of lab-wide data dictionary, there's a process for us going through it and making sure that we get the numbers right. We started pre-printing all of our, so I'll move on now. So now we pre-print all of our peer review manuscripts. It's kind of just habit now. We've gotten over that fear. I think there was a fear among a lot of the PIs that they would be excluded from submitting to journals if they did this. You know, it's kind of like prior art concerns. And we explained that's really not the case for the good journals. They've kind of bought into the preprint system. We try, again, it's not always appropriate, but we try to publish in open access in all of our manuscripts. And then the last two things, I think that some of the biggest benefits were we containerize all of our minimal preprocessing now. So from raw to minimal preprocessing is completely done with fMRI prep. And then we also run MRI QC on all of our data. We haven't officially started using any kind of machine learning tool to exclude include. That's on the list of my projects. It wasn't part of the original goals, but I've learned more about how useful that could be to kind of automate the process of inclusion exclusion. So we're working on rolling that out now. So we do run MRI-QC on every image that comes to the center. We're not actually using that in the way it was really intended, which is an automated system. But that's on the list of projects. And so conceptually, I guess the takeaway from that, so that was the mapping process. That's what we achieved. And when people, so some of the things that have come out of this that we conveyed in the paper that we published was when, you know, I get asked a lot now that people have read that paper, how would I do this if I wanted to transition my lab? My wife asked me about it since she's a PI at UAMS in cancer, like what would it take? And so these are my four kind of takeaway, kind of my elevator pitch for open and reproducible science is these four things. That's what I've come up with because I get asked quite a bit. So find your workflow, and it does differ. I would think that all workflows would be the same, but they're really not when you start thinking about animal research, you know, like wet lab. It's very different than imaging. So identify your workflow, specifically write it down in like an engineering box diagram with causal arrows and whatnot. And then read the literature, you know, start with, I guess, the paper we just published as a way to kind of quickly digest some of the terms and whatnot, and get that paper, by the way, has a huge number of footnotes. I can't believe they actually let us do this, but we footnoted like all the tool URLs. So like right in the footnotes are like the links to ReproNim and OSF and all that. They let us do it. I was thankful. So, you know, read this primer paper and then read each of the papers cited in that paper to really get a sense of this is what reproducible and, you know, open science is about. And then I can't stress enough, identify a data standard in a field, or if you look into it and you can't find one, find the top people in your field and see how they do it, at least. So, you know, there's going to be some proto data standard because there will be one eventually, and you don't want to have some completely orthogonal data standard that you're using when the top people all agree on, you know, some X standard or something very close to what they're already doing that it's easy to map to. So find your standard. If it doesn't exist, find a proto standard that people think is reasonable. And then kind of follow this template where you just map the practices onto this workflow using the data standards. It seems to be the recipe for, you know, low cost, high benefit success. Okay. And so this graph, so this is, I actually pulled this figure directly out of the manuscript. So one of the things that we thought would be most beneficial, being the size of the lab that we are, given the resources that we had, we thought the biggest impact that we could have is not just pointing people kind of in an abbreviated, summarized way of, you know, what the tools are, what the tools do, but we really wanted to give people an assessment of this is what it's going to cost you, and this is what we think it's going to benefit you given our circumstances. And so before I was talking about, and I make this much more clear in the manuscript, but before I was talking about these four principles and 4 tool sets, but those tool sets aren't necessarily best implemented as blocks. So part of the roadmap that I described earlier was breaking down those tool sets into digestible work chunks, steps, we're calling them, that map onto those tool sets. So you can see here in the category, I have the 7 steps that we actually went through in terms of deliverable work units towards the goal of being what we considered a win on transition. And I'm mapping them onto, this is the category of tool set that they come from. And then I give a brief description of this is what we actually mean by it, and then cost benefit. And these, again, we didn't think about writing this manuscript until we were done. So this is all based on hindsight, which is, probably biased, very much so biased, but it's hard. We were trying to do our best to remember from, I had taken notes, I'd looked at emails, trying to think, what did this actually cost us in terms of FTEs? We really think about it in terms of FTEs. And then what we think the benefit is. And the benefit really is something that was, at the moment we finished, we'd already kind of seen the benefit. So we had a pretty good estimate of that. So let me walk you through. So the first thing we did that we thought was the easiest to do was, and this is primarily because there was a computer scientist on staff. So this is one of the things I want to kind of reiterate several times as we finish up. I'm a computer scientist, I'm a pretty good programmer. And so some of the things like using GitHub and migrating a software stack from MATLAB to Python was very easy for me to do. I was already fluent in Python anyway. I programmed Python on my own projects at home, very fluent in all the tools. It was very easy for me to basically just rapidly duplicate our software stack in Python. And it's much better because actually, I like the both Python language better. I think the tool sets are better. So our software stack was transitioned rapidly and probably ended up being more effective, better curated because I was starting from scratch. So we consider that cost low. I think that cost is only low because of the unique circumstances of our group. I think other groups that might not have that kind of personnel would struggle a little bit more. And we make that caveat in the paper. But the benefit from doing that is extremely high. Just in terms of, shared projects and reusability, and I think just linking code to manuscripts should be the de facto standard. Like the methods and sections are just not good enough anymore for how complicated the processing that we're doing. So you really need to be able to just click on a link, bring up, a GitHub, and just trace through tricky sections of analysis, to make sure the person's doing it correctly. I think there's no limit to how beneficial that will be in the future, particularly for trainees. Seeing how a text description of a method translates into an actual software implementation, particularly if those software implementations are written by really good programmers. It helps on a lot of levels, not just correctness, but learning how to write good code. So that was the first step. The second step, we thought that was low-hanging fruit, but it was primarily because I was the architect of the process and I thought that was the place to start. It's probably higher for the groups. That's the takeaway I'm trying to give you. And then the second step is pre-printing. This is such a no-brainer. It's hard to believe we didn't do it. It was really an anxiety about giving your thoughts away before they were published, right? But the reading suggested that that's just not a concern, and we've, there's an anxiety to it that's unfounded. So it was very easy to do. Essentially, when you have your manuscript done, before you format it to submit for peer review, you just make a PDF of it and submit it to SciArchive or bioRxiv, whoever would accept it. Very low cost to do it. In fact, they make it easy to submit manuscripts directly from the repository for some journals. And the benefit of that is that we get to share with internally in the lab, externally. It's there's just a place where the summary, the work exists that we can share publicly and discuss it. And it's timestamped, so issues with getting scooped and whatnot are alleviated. So the biggest thing, and I should have highlighted this in red. We didn't put it in red in the manuscript, but I thought about bolding and reading it. Is that the most important single thing that we did was map to bids. There's no question. Every lab should be doing this regardless if they want to do open and reproducible science. There should just be a national call to map all data to bids. And if a lab is not doing it, really is unacceptable, given the benefits of doing it. So so training the research coordinators to help me do the coding was a big lift. So I had to train people in Python that had never coded before, and that was a long process, just educating them. We didn't have any other programmers in the lab. And then actually mapping individual research projects that were stored in custom formats with custom naming conventions, typically coded by people that were not professional computer scientists, so they didn't use necessarily best practices for naming and reusability and whatnot. All of those individual studies, and I think there were 9 ongoing studies that we decided to map. All of them had to be individually mapped into the BID standard. This was a huge lift. It was by far the most costly part of the process. But again, it was also the single most beneficial thing that we did, because it gave us access to repositing, BIDS tools, data dictionary integration. It also gave us access. It made it clear that when you write second level software analysis, right, for like your task design, for building contrast and whatnot, if you have data in BIDS, software naturally starts adhering to good software practices, because everybody's interacting with the data in the same way. So software naturally becomes reusable across PIs, because they're looking at the same data structures. And so that was just a natural thing that came out of using the tools, or mapping to the BID standard. So it's made it a lot easier to write custom software for PIs, because everybody knows what the data is going to look like going into it, because it's curated correctly. So once we had that in place, the next step was we just implemented the next day, ReproNims, or sorry, fMRI prep. So once we had it, we started using fMRI prep right away on any study that mapped and validated. We immediately started using that tool, as well as MRI QC. Very low, as long as you're in BIDS. It has to be in BIDS. It's very low to do once you get over that hurdle. And again, public reposition, as soon as I had data sets mapped that were, they were validated in BIDS, and the consent form allowed us to do so without any questions whatsoever, we immediately put on Open Neuro. Studies, and just a caveat there, studies for which there were issues about consenting that we were concerned about, we are doing our best to migrate intermediate files to OSF. So rather than publish, ROIs and tables and appendices, just give everybody the entire statistical map, throw it on OSF, make it available for people. It's very easy to do once you understand the OSF ecosystem as well. And then, like I said before, the FAIR data, the data dictionary mapping was by far the single hardest thing, other than the BIDS mapping, but it was the thing that would not have gotten done. Again, extremely high barrier to entry to build a data dictionary, and the benefits of it are really not proximal. It was just an exercise where we had manpower. We had skilled manpower with nothing to do, which is how we got it done. We would have not got it done. Very clearly, we would have not made that step successful without COVID. So it's just too high a bar, and the benefits are too diffuse and kind of in the future. We're glad we have it. We feel very fortunate that we were given the opportunity to do it. But that's something that needs to be thought about in terms of getting over a hurdle like that. It's very challenging. And there might be things that we don't know about how to make it easier. But that was, from our chair, the least cost benefit, favorable ratio of all the things that we did. Everything else is really like plus EV, other than that step, or neutral. And then the last thing we did, just because of the nature of a transition where you kind of have to pre-register when you start, almost all our projects were long-term funded projects, so we could pre-register things that were already collected. And so the last step that we implemented was actually all new studies or all new secondary analyses. We're actually we have established policy in the lab that you pre-register, even minimal pre-register on like as predicted. But PIs should be pre-registering anything that they're going to do that are part of the imaging center. They have to do it first before they actually, do it alongside your IRB protocol submissions. Just get it done before you actually start collecting data. So those are the things we implemented. We completed March 2022. I think the takeaway from everybody in our group was that this has been a very positive experience. Most of the negative experiences were borne by myself and the 2 coordinators that had to do some of the custom software mappings and dealing with data formats and log files that made no sense, and trying to figure out how to get all that mapped appropriately. That was a really heavy, frustrating lift. One of the things that came out of the process now is PIs are aware of how they should format their data ahead of time. Ideally, they write log files in BIDS format, so it comes out of the presentation machine correctly. We don't have to do any kind of mapping. But the overwhelming response to the lab was that we're very, very happy that we did this. We definitely think that we're more productive now that we got through kind of the bottleneck of all the upfront costs. I guess that the thing that's most frustrating stuff for people is pre-registration, because it's just when you're busiest, it requires extra work, right? You're writing the grant, you're writing the RB protocol, and you have to additionally kind of have to write the journal paper, everything but the figures up front. So there's grumbling about that, but we kind of put process in place where we work together. The data team works with PIs as they're creating studies to make sure that they're doing everything smart up front. And so the last slide, right on time, this is perfect. So I wanted to kind of flip the presentation a little bit and just posit some questions to this community, because you all are the experts. You know, I know these names, the people kind of in the Zoom box, right? You're the authors of all of the most important work in this field. And so I would posit to you to think about this case study as a customer experience, right? So the Burke in 2018 was a completely naive customer that had one advantage, is that we had kind of a full-time computer scientist in the lab. I do neuroscience research, but I mean, I'm a dyed-in-the-wool computer scientist. So some of those barriers of dealing with all this technical documentation and coding and, you know, changing the stack, those barriers are a little bit lower than average. But think about this. So if you think about us as a customer, with that caveat of having that expertise on staff, how would other centers, if you're thinking about kind of promoting your product and getting uptake throughout, you know, the broader scientific community, how would other centers of comparable size and resource do? And you know, what would their cost benefit map look like? Because you could think about this table as the user experience. If I was filling out a survey, if I just purchased a new car, they're constantly harassing me for the user experience. Well, the table is the user experience. And if you're thinking forward about how to increase uptake in the community, I would focus on the table on the right and like, how do you lower costs? How do you increase benefits? Or, you know, maybe the benefits you can't really control, but you can at least control the costs, right? In terms of thinking about how you curate your materials, how you pitch your materials, you present the materials. Because I think our lab is a good use case. We were completely naive. We didn't know where to start. So we did what we did, and this is the outcome we had. And these are kinds of what this is the experience we had. I think that might be useful for the community to think about projects moving forward. So I think I don't have any other slides. I'd be happy to answer any questions. I'm curious to how this is going to go in terms of questions, but I appreciate your time. I hope this was useful.
That was fabulous. Thank you so much. I'm going to, let's see.
And one caveat, so I'm hard of hearing. And unfortunately, when I do Zoom, I only get sound out of my laptop speakers. So it's quiet. So if you speak, could you speak pretty loud so I can hear you?
Okay, thank you. I will talk as loudly as I can. Dorota says in the chat, awesome work. And I see Yarek with a question. So let's jump right to Yarek.
Okay.
Can you hear me?
Yeah.
Awesome. So now when people ask me, like, describe with one word what you know about Arkansas, I know that that's the success story, you know, because that was exciting. Although I missed beginning like 15 minutes, but what I've heard was really encouraging and really informative. And for us, as you mentioned, all these, you know, metrics for benefits. First question, like you point that, let's say, step 3, fair data, map data to bit standard. You say that the cost is high, right? And in your case, you were converting data which you already collected, which is indeed, then it's more like archaeological expedition. You know, you need to discover the detail and you need to map it, and everybody wouldn't know, did it differently, right?
Yeah.
But what we do within ReproNim, we also encourage people to think about it ahead of time. Let's say one of those is ReproIn convention, where people agree how to name their sequences in the scanner once.
What is that? Can you spell that? ReproIn?
See, exactly. Yes.
ReproIn?
Right. And I'll point you more specifically to it. We have, and we did that starting from 2020, maybe, or 19, when we've got new scanner, especially when you get new scanner, that's the best time. And then everybody just knows, and it's pretty much like bits. You name them as bits in the scanner, and then conversion is not flawless, but it's much more streamlined. Pretty much I can convert the entire study without interaction unless humans screwed up, right? If they make a mistake. So our point is to minimize human interaction. if you could make them type as little as possible, then world becomes a better place, right? And usually they screw up anyway, like they enter capital O instead of 0 or something like that. That's drives some people nuts eventually when they cannot even type the number. But the point is that by minimizing the entry and making them invest those 5 maybe minutes in the beginning of the study once to name those sequences according to some convention for the entire center, it just streamlines conversion later on. We did that, then we distributed it through our postdocs to other universities. Let's say at Princeton, they convert many studies that way. And so I would recommend to look in that.
We did, so when we, so I don't know if you have familiarity with Philips. So our Philips scanner, the DICOM header doesn't like have, like, there's no way to like create, at least we were unable to create like the information automatically for like a JSON file of like all the parameters. So that stuff had to be reconstructed like by hand. Our new Siemens scanner, when you do a DICOM to NIFTY conversion, like all the info, like some of the things that are like really frustrating about the mapping process went away because it just like automatically generates it. We had a lot of trouble mapping. The old Philips Achieva DICOMs are just really not compatible with pulling out the information we needed. That was one of the primary sources of frustration we had. From the scanner itself, I would say. So we have invested a lot of time up front because we just cited a brand new scanner like in March. It was I think that was when it was actually turned on for production. We took the time to get all of the naming of all of the sequences in a standardized format across all the PIs. We have our MRI physicist, Brandon, she's a real stickler for proper science, and so she kind of runs the scanner now, and we don't have any say. So I got to her, she bought into this and kind of enforced those kinds of standards internally, which is nice. So we solved some of those problems. I agree, it is a lot easier if you do it up front, like when you have a new scanner. 100%.
And as for Philips as well,
Philips, and just in terms of like the sequencing, so we actually like the actual slice sequences are very.
It still doesn't. It still doesn't give it, right? It still doesn't give it. It's just there is heuristic. There is a script which I wrote a while ago, and Chris Wharton kind of promotes it now. This is the way, But it doesn't work for multi-slice acquisitions. But at least for regular interleaved and whatnot, it works.
Okay.
So you could figure it out based on UID. You would still not have time slice timing. So you would just evenly divide it within the TR. But at least you can figure out the order. That is already kind of good help, right? But on that note, I will not tell the story, but on that note, I want to recommend to talk to projects or manufacturers which are affecting or which you could improve. So in your talk, I've heard a lot of consumer talk, right? So you use this, you use that, but I haven't heard anything how you interacted maybe with those projects which kind of you started to use. In my case, like first thing how I start using project, I look at the source code and see maybe I could just chime in with some improvement or some complaint. Quite often that happens. And in your case, let's say with Siemens, I would have just let tech personnel know that we need slice timing. And just even though if it goes into the void, but maybe some pursuant client would finally pursue them to put that information in. Actually, I should, which reminds me, I should pursue on that project as well. But the same about bits, right? So you've used successfully, I haven't got the detail besides that you use ReproBits, Dave's work, right? So using those terms to put into bits, it's super useful to know, right? That you succeeded and you did it for some data sets. Are they? And you said that they're all on Open Neural. I'm yet to find them.
Well, there's so primarily my studies are on Open Neural just because I had written my consent forms. I was kind of like the newest faculty at the time, and so I wrote my consent forms thinking I had to do this because I wanted to make it public. So our other projects are not the consent forms the way they're written. It's ambiguous whether or not this is okay. And so that's something we're working on. Like we have the data ready to go. It's just a matter of even de-identified. I mean, when you have some of the psychopathologies, there's issues, right? It's when we're recruiting from a very geographically isolated area, these things can be de-identified. So
As
that's
for
good stuff.
wording,
Yeah, so if you
as for
search my
wording,
name in Open Neuro, you should see all my stuff is there.
Uh-huh.
And if it's not there, it's on OSF. I've made all of my work is public as I can make it legally. What was it?
And for the wording for the next studies, did you suggest them with the wording? Do you know the wording?
Yeah,
We
so
have another project.
we're so like what you're saying. So like because we've done this now, we're putting in place processes during study design. Hey, what are we going to name? Like just working with the PIs to say like this. Tell me all the data you can collect. This is how you should be naming things. This is when if you have a graduate student writing log files out from PsychoPy. Hey, let me talk to them about their naming structures just to make sure they understand. To name it, just name it correctly right off the bat, put everything in tab separated value format right out of a log file. It's so easy to do in code. It just causes friction down the road if you don't do it that way. So like, even internally with 6 people, it's an education process to get everybody on board because I mean, I'm presenting this like we're a unified front. I mean, there's still frictions inside the lab of people value this versus other people value it less, but at least everybody agrees to pull in the same direction. But I'm by far the most pro this, and other PIs are less pro this, but we are pulling in the same direction, and it's an education process internally. Yeah.
Let's get a JB
I'll
question
press on
in
to it. Yeah.
as we come up towards the end of our hour.
Yeah.
Oh, yeah. Thanks so much for the feedback and it's just very uplifting when you see something like that succeeding. I think if I look at other brain imaging centers, sometimes there's much more history and larger groups and folks have their ways of doing it for years, and so the barrier sometimes are much higher, and
Yeah.
So I was wondering if you just had some kind of a thoughts on how do we do it in a much kind of a like a bigger it requires so much work even in those good conditions. Is it feasible in a bad condition?
Yeah. That's weird because I would think about the other the big institutions have all the resources, right? And you have undergrads with so UAMS doesn't have a co-located undergraduate facility with computer science majors that you could maybe pull off to get people that know how to code. The limiting factor really for us is having people that understand specifications, formal specifications like bids is really hard for somebody that has never seen one before to digest. It's educating them and then understanding how there's like this seamless representation in code to what's in the specification. That's a big lift for somebody that was a biology undergrad that has been doing, human subject, they skid and do assessments. That was the resources that we had, and they were willing to learn. It would have went faster if we had a few really good undergrads in computer science that I could have just given the spec. They would have figured out in a few months and coded up some solutions. So I would feel like at bigger institutions, those are more likely to be available. And I would, maybe a whole, I don't have any, I really haven't thought about it, kind of flipping it like that. I would push for, NIH to basically create grants to just do this. It's kind of like unlocking all this institutional investment by having teams kind of go into groups and do the mappings for them, just so they can keep doing science and not worry about this. I don't know if that's on the table. I'm not involved in any kind of higher level discussion.
That's true. I mean,
I was talking with Dr. Kennedy before we started this, like that push by NIH to kind of require this might unlock some it'll at least put incentives somewhat in the right places to push people. And maybe that tied with some money might eventually do this. But again, the Berkeley, so the other thing I should have said it. The Berkeley is a brand new, not brand new, it was started in 2008. So we only been around for 14 years. So we don't have an enormous historical catalog of imaging data and we didn't, and then the other thing maybe I didn't make clear, we did not map everything that we have. We only mapped studies that had just completed, where all the people involved in the study could explain to us what everything meant, or studies that were ongoing. we didn't try to dig back into the archives of closed R01s where the postdocs and graduate students that worked on it are gone, and it would be almost impossible to figure out what was going on. We didn't do all of that work. So if that wasn't clear. So we mapped, I think, 9 total studies, and we are prepared to map 2 additional studies that we have funding for, and they're starting up. So we're trying to do everything correctly, pre-registration, writing the log files out correctly, getting all the naming correct, getting,
as Jarek was saying, the prospective aspect is really important. Like, you'll be so much more efficient in this respect.
Yeah. And again, this could only be unlocked by our center director has an endowed chair, and he was willing to part with a good chunk of it for a few years to fund this. That was really where the money came from. He saw the value and did it. So much of the credit goes to him for believing me when I said this needed to happen.
Thanks so much again. That was fantastic, Keith.
Thank you.
I'm so happy you persevered in making that happen. And not that I would wish the pandemic on anyone, but again, it's interesting to see how that played into being a little bit helpful.
I'm willing to stay on. If people want to ask more questions, I mean, this is, it's great if people have questions, I can help out. I'd be willing to stay a few more minutes if that's okay, or if there's like a hard deadline to close this.
Not a hard deadline for us. So if you're available.
Yeah, I could take a few more minutes.
Yep. Felix.
Yes. I see a hand up. Sorry, I don't know your name.
Felix.
Yeah.
Okay.
Yeah. Hi. Great talk and really nice overview how to basically get there, because I saw a lot of overlap like 10 years ago, let's say, in our lab. One question, how, like, as well for others, so what are the pieces that you could have not done at the beginning to basically, and still would arrive at let's say half the way, if you will? So what I see here, if you do bits, you have a lot of the other stuff already.
Yeah, do bits.
But fair data, like the data dictionaries, is one of the unsolved problems in my view. But and so are there pieces of your, let's say, or categories that you could have, let's say, delayed or not pursue directly, and that might be as well like basically ranking for people who don't have basically can implement the whole plan directly, but can start like slowly? What would you recommend?
Yeah, so that's a great. So we didn't need to migrate our software stack. So that's kind of like a, MATLAB's a proprietary language, so it's I mean, in the strictest sense, it's not open science, right? Because there's a big part of the world people can't afford to actually buy the licenses to run the code. So it just in terms of usability, migrating to Python is good, but it's not necessary. So if you didn't have someone that's willing to rewrite the entire center's stack, but I had always been wanting to do it. I'd been wanting to do it for years just because I hate MATLAB and I wanted to use Python. So I kind of I might have double dipped on this and used as an excuse to migrate to Python. So you could definitely push off on doing that for sure. The amount of money we save in licenses it would take years and years to cover the salary costs that were incurred in me migrating the stack. So from a money standpoint, it wasn't a win, but in terms of, it's a better language and it's developed more vigorously and it integrates with a lot of other things, a lot of other tool sets that are out there. So in the long term, it was the right choice, but you could definitely, of all these things, you don't have to migrate your software stack to R or Python. I just, you do that for other reasons. It's just a better decision. But in terms of open reproducible science, it's not necessary. It's encouraged, I guess. That's how I would look at it. I don't know if the community feels that way. Like, is MATLAB kind of verboten, or because it's technically not open science, right? So that's part of the requirements, right? It basically has to be free, transparent. That was my impression.
There are some workarounds for compiled MATLAB, but in general, MATLAB tends to be a barrier to openness if you're trying to stay as open as possible. But it's not completely issued, and SPM and things like that are good participants of the community. But again, a lot of it gets pushed to Python and R and things like that.
Yeah.
So I know we're up on the end of our hour. The one comment I wanted to make, and as you foreshadowed in your table, that this low benefit perceived, at least short-term low benefit on the data dictionaries is a problem, because of the complexity there. You are correct in identifying the long-term benefits is where those, may be observed. But again, that's still kind of the cutting edge of, how do we do things, that, help people realize those, long-term benefits, and really, so this sort of the ability to create these semantic markups, create your own personal lakes of data, and combine your lakes of data with other people's ponds and lakes of data, and really be able to integrate data better in future studies is really sort of where we're trying to push and get tools and examples where we can begin to at least see where that future benefit will be, if we can get enough content in there to be relevant. So again, thank you for illustrating that and admitting that there might be some long-term benefits there, but again, we're still all kind of a little bit short and really selling that part of the mission very well. Again, so thank you very much.
Yeah.
It was great to have you present this. It's great to have had you do that. We are working on these, annotating who these sort of Repro Center type places are. So we are pleased to, if you will let us to put you on our sort of list of good examples of these kinds of things for this effort. So for the record, for those of you who are still here, next month's webinar will be on Friday, January 6th. I will, and some of the other senior folks will be doing a state of the ReproNim presentation, just to sort of an annual update on what we're doing internally. And then the rest of the spring semester will be filled with more examples from the community. So January 6th, 2 p.m. Eastern time, myself and the ReproNim team will be talking about the state of ReproNim. So again, thank you, Keith. Keep in touch and let us know how we can be helpful. And it was so great to hear you.